Hello, my name is Diego Luna. I am a bioinformatics licenciatura student from the National University of Entre Rios, Argentina, and I'm working with Dr. Gonzalo Parra and doctorate student Maria Freiberger on local frustration footprints in the Globins protein superfamily. In the protein folding process, the internal energetic conflicts are minimized until the structure reaches its native state. But in order to achieve functionality, some of the conflicts must remain. These local conflicts are related to important roles in protein, such as allostery, protein binding sites, cofactors, ligands, and catalytic sites. Energetic frustrations can be localized and quantified by comparing the contribution of the extra stabilization energy from a given pair of amino acids in the native protein to the statistics of the energies that would be found by placing different residues in the same native location or by creating a different environment for the interactive pair. If the pair is more stable in the native configuration, the contact is called minimally frustrated. If the stabilization lies in the middle of the distribution of alternatives, the contact is neutrally frustrated. And if the native pair is less stable than the other possible ones, the contact is highly frustrated. We use the frustratometer to calculate the individual frustration of each structure. Conserved frustration patterns are detected by calculating the frustration information content over the frustration results that match the homologous residues guided by a multiple sequence alignment from each family. We know that local frustration is related to functional aspects in proteins. This triggers the question, can we detect functional adaptations in proteins by analyzing differential frustration patterns in related proteins? In the seek for answers, we chose the Globin family as the research object. We want to study globins to detect different functional adaptations in the different branches of the phylogenetic tree of a good part of the superfamily. Until now, we've studied three families: alpha globin, beta globin, and myoglobin. Families which have a very similar structure. The workflow goes like this. We obtained a non-redundant multifacet of 40 alpha globins, 41 beta globins, and 21 myoglobins, align them and handmade curated the alignments. Then we frustrated them individually with frustratometer and in a whole family level using the evolutionary frustration pipeline, obtaining identity logos and frustration logos. We weighted the contact frustration information content, obtaining the interchain contact maps of the families. To study the important residues of the myoglobins and the residues linked to pathological mutations in alpha and beta globins, we included the contacts between chains and frustrated the data set again, obtaining remeasured and filtered contact maps. On the other hand, we searched for theoretical and resurrected globins, frustrate and compare their structural distribution of frustrated contacts with the extant ones, finding similarities with each correspondent set. We started with alpha and beta globins. We observed that some of the residues changed between families, and with them it changed the frustration states for the given position. For example, the position 127, we can see that this histidine in alphas doesn't have the same frustration state than the glutamine for the homologous position in betas. For the position 39, the example is much easier to see with glutamine and lysine. In previous work, we saw that there's no correlation between identity change and frustration state change. This is our start point. Do these contexts respond to evolutionary adaptations? With these results, we weighted the context of the whole family by contact frequency and observed similar patterns between the contact maps of each family. These patterns are associated with helices or specific sites that are distant or close in sequence, but at a structural level, they are close and interact in a minimally frustrated, very rigid way, or rather highly frustrated, more dynamic way, acquiring some function in this later case. Then we got the myoglobin cell to process. We obtained the contact maps and saw that the frustration patterns were still there. We observed that alpha globins are more frustrated than beta globins, and that myoglobins are less frustrated and have more variable frustration patterns with few distinguishable high frustration patterns, which translates to low frequency of the same state of frustration for each of the same contact in the entire family. When we filtered the contact maps by structural and functional importance in myoglobins, we found similarities with the contact maps from alpha and beta families when we filtered them by only having in count the residues related to hemoglobinopathies. Here we can see the frustration results of the three ancestral globins of each family. Since these families share a common ancestor, the differential patterns in this data set may correspond to specific functional adaptations to the activity and context in which these proteins currently operate. Until now, what we got is a hint about where and what to look at. This ancient globins shows us a node backwards on the history of this family, and let us know in which point the branches could have separated. And by including new families, we believe that conserved patterns can be used to explain the evolutionary history of protein families, to detect specific functional aspects of them, and to better understand the relationship between sequence and function at evolutionary scales. The concluding remarks are that the myoglobin family is minimally frustrated, showing that it is structurally a very stable family. The frustration patterns of contacts related to hemoglobinopathies show similarities to the patterns obtained with the important contacts in the myoglobin family. This explains the hemoglobinopathies phenomena in a structural point of view. Ancestral globins show similar frustration topological distribution with the extant examples. The frustration contact maps of evolutionarily related protein families show patterns that are evolutionarily conserved. These patterns show variation in individual contact frustration states and frequency, but an almost identical topologies. For the future works, we we believe that with more ancestral structures available, we will obtain contact maps to relate the extant ones with them. We have to model neuroglobins, and we are considering truncated, fatal, and cytoglobins. Thank you very much.